Wonderful introduction, and thank you, Victor and Ankarnita, for your leadership in the area, and friends for having me here. I'm so excited to come here. This is such a neat community coalescing together around software, pipelines, and tools. And so I was really excited to come and share what we do. We typically work in the structural space, and as I said, I'm director of the Rosetta Commons, which is a very similar community to this that comes together around software, historically around structure prediction and design of biomolecules. So yeah, I'm really excited to be able to share with you today. And thank you, Justin, for the introduction of the machine learning methods. I'll do a little bit of that too, but it's good to have the foundation. To start with, as I was looking at my talk, I've had that picture of an antibody for a long time on my talk slides, and it's back from when we did physical modeling and models of different H3 structures, and so I wanted an update. So I took my talk title and I fed it into DALL-E, and I asked DALL-E to make me a new title slide, and this is what I got. And it's a good reminder that these AI tools are spectacular, but also sometimes really not quite with it, right? We train it on some distribution of, say, images, and it learns that letters go together, but it doesn't actually understand the letters and know how to put them together, right? And so I think this is I don't know, I'm gonna share some stories of things we've been playing with, but also a little bit of a warning about these tools. Here's a related one. That figure was actually accepted for publication in Frontiers, and then as soon as Twitter found it, it was retracted, because obviously that image and all the other figures in the paper were generated also by AI models. So they can be very convincing. You can convince editors and reviewers to publish papers. So similarly, I want to think about if we're going to make generative models of AIRR data, I don't know, there's a sequence. Is that a real sequence? Is that not a real sequence? So I put a poll in the Whova, so you can vote later, but I don't know. How many people think that's a real sequence? Okay. Okay, how many people think that's not a real sequence? Okay, and are you just guessing, or does anybody have any evidence, like things you're like, oh yeah, that's wrong? This and the W look good. Yeah, EVQ, there's a lot of familiar features. Okay, so I'll let you vote in the Whova app, and we'll come back to this later, but this is a little bit of a thought question. If somebody came around to your data commons and dumped in a few 100 million sequences that they just made up, would you know? And how would you know?
We'd take them.
You'd take them. I guess we'd have to make up annotations too, and yeah. So we've been thinking about that, partly because we've been training these language models, and in preparation for this meeting, I'll show you some other papers I've been reading from your community and the literature. I don't know, to be honest, I was a little bit anxious about talking to this community because we're dealing with the sequence data, and honestly, I don't know the answer to this question, how to tell if it's real or not. So, okay, so what do I typically do? Typically, my main problem is protein-protein docking, and we spend a lot of time applying that to antibodies. So in antibodies, that means first I take sequences and make a homology model of the antibody, which is, of course, is a dumb, easy thing because they all look the same. But what I really want is all of those complementarity loop regions to be exactly right, so then I can take it down, because who cares about this? What you really care about is how does it bind to the antigen. So I'm going to take those structures and then either have a structure of an antigen or maybe predict it and then try to figure out how these things bind, what the epitope is. Could I make it bind tighter or weaker? Could I stabilize the antibody, make it behave better? So we've been doing this for 15 to 20 years maybe with Rosetta-based tools, all based on physical representations, X, Y, Z coordinates, and energies as best that we can calculate it. And then I had a summer intern in 2019, so that's just pre-AlphaFold, right? And in 8 weeks, he could do the H3 prediction as well as we could do it after 15 years of using physics. So we've kind of skipped over, since 2019, the whole lab has kind of shifted into various deep learning-based methods. And we've run the gamut of all of the tools that Justin put up there: convolutional networks, multi-track attention models, language models, graph networks, diffusion models. And it's just been fun for us. I'm not sure we really understand all of this because we're engineers and physicists coming in using these tools, but we've been playing around and trying to test them and see what they do for us. Today I'll have time to talk to you about a few of them. I particularly chose the ones that had to do with antibody sequences. I don't do any TCRs, but antibody sequences and things that might relate to the AIRR community. Before I go further, I'm director of the Commons. We do license our code commercially, and the money comes back, kind of like the group that Victor just talked about. It's got a research community. The money comes back in, and we spend it on the research community for the common good. And so there's an institutional conflict of interest there. Cyrus used to give out a GUI for Rosetta, and then some of our new algorithms that one of the students wanted to spin off a company, so we did patent one of the algorithms and license another one. So there we go. Okay, so 2020 was the big breakthrough that shattered, that really changed a lot of how we looked at things. And this is, hopefully I'm not stealing Charlotte's thunder with her group slide here, but this is groups getting better and better in CASP. This is Rosetta getting better and better, and then saturating, and then CASP 14, AlphaFold, does really well, and everything is kind of, I think Mohammed Al-Qureshi said the protein folding problem is solved. If you should get out of the field, the rest is engineering. And of course, I'm an engineer, so this really just opens things up. And I think just like the human genome was solved in 2002, then it was again solved last year. I think there's a lot of work to actually know what's going on with these structures. So in physics, we would start with the physical laws, and then we would do some kind of calculation or simulation or search and predict some kind of phenomena. In our case, it would be structures of, say, an antibody bound to an antigen. Then we'd compare to a crystal structure, some kind of experimental data, and then we'd go back and say, okay, what did we get wrong? And, well, we probably wouldn't change the laws of thermodynamics, but we might look at the Coulomb formula and say, I don't know what we really know what we're doing with a dielectric constant. Maybe there's a better model for the dielectric, and what does that look like? And then the last 5 years, we've been playing these machine learning games where instead you start with a big pile of data. Data Commons is a beautiful place for a big pile of data. You split it into features and labels in the supervised case, and then you put some network in and you go forward and backward until you reduce your risk or your error with what you're trying to predict. So I don't have any models of, any physical models, any explanatory models, but I have a way of putting these data together, and it's super powerful, as AlphaFold showed us. So let me start with the first question I want to address and talk to you about, which is, could we use deep learning to predict high-resolution structures of antibodies? Again, this is something we were doing for 15 years and with physics before we started the machine learning part. And why would you want to do this? AlphaFold 2 is wonderful, but it relies very heavily on multiple sequence alignments. So it needs to know this residue and this residue mutate together, and therefore they might be close in space. And if you take away the MSA, it's much less powerful in its predictions. And this community knows very well that antibodies and antigens evolve over different time scales and in different places, right? So first of all, your AlphaFold doesn't have access to the repertoire showing the evolution of the antibody unless you feed it a repertoire, and I'm not sure if anybody's tested that yet. And second, between the antibody and the antigen, those correlated mutations, the antigens mutating over months to years, and you just don't see those patterns. So it's at a real disadvantage with its core tools. This has been shown. Brian Pierce's lab did this nice study of docking of antibodies and showed that if you took 5 AlphaFold models, maybe you'd get 40% acceptably correct when you dock them together. So it's well known that AlphaFold struggles with antibodies, and I think T-cell receptors are the same. What about AlphaFold 3, which came out just a few weeks ago? The preprint claims that this number bar comes up to 60%. They get antibodies and antigens docked 60% of the time. There's some fine print. The normal predictions, they make 5 models, but for antibodies, they make 1,000 models, and then they pick the best ones by their ranking criteria. So it says you've got to sample a lot and search to find the right one, because it doesn't necessarily get there. And by the way, the server, we don't have access to AlphaFold 3, so the server I can't tell because it doesn't have a switch on it, but it doesn't seem to detect whether it's an antibody, so I think it's making 5 models. So I think if you use the server, you're not going to get this 60%. You're going to get a much lower number. And we're testing that 20 structures at a time, or some people are testing that. And by the way, the accuracy, they still show, they claim that they rely less on multiple sequence alignments, but the accuracy is clearly still tracking with MSA depth. So I think there's still a need for, physics does it just from the sequences and puts things together and doesn't have to have all the evolutionary history. So I think in principle, you could make a machine that can dock proteins together with physics. So we're still playing with tools to try and head in that direction as a goal. All right, so where did we start? In 2019, we used convolutional neural networks, which operate on images. And so we would take a structure and look at the geometries between 2 residues. So if you took 2 amino acids in space, what's the distance between them and what's the orientational components between them? And then these images, this is residue 1 through n and 1 through n, so these are distograms, and then the other layers would be the different angles involved, so you could get 3-dimensional coordinates, 3-dimensional orientational relations between 2 residues. And I really like this because it's machine learning, but it's learning what it should, is how does an alanine and tryptophan interact? What angles and what distances do they fit at, according to how often we've seen them in the data bank. So I really like this representation because it should harken back and learn some physics. Whoa, holy moly cakes. Let's start over. Hang on, I don't know what happened there. Okay, so we make a neural network that takes the sequence, predicts the orientations and distances, and then at this time in 2019, we fed them to Rosetta, which by the way was what AlphaFold 1 did, is it fed things to Rosetta, and Rosetta solved the structures. That worked pretty well, compared to other methods. And one of the surprising things that came out of it, we're just predicting structures, but one of the new things with machine learning is you can predict, but you can also give a confidence value. How confident are you in that structure? And for kicks, we took our confidence measures and we plotted it against an actual functional characteristic of an antibody, which is how tightly it binds. This is a data set from Sorel Fleischmann's lab on an anti-lysozyme antibody where they'd done lots of mutations. And so we put all of the different mutants in and calculated these confidences and then plotted it against their binding enrichment relative to the wild type. I didn't expect anything out of this because why would my structure predictor know anything about the antigen? I didn't train it about antigens at all. And surprisingly, it's not a one-to-one, it's not a correlation, but there's an enrichment. If you take the things we're confident in, there's a big enrichment in the number of things that bind tighter. And in fact, if you look at the top-ranked mutants, they all improve binding affinity. So that was really intriguing to us. There's a little bit of caveat here is that we're using positions that Sorel Fleischmann identified because we thought the problem was really hard at this time, so we took the helps that we could. We've now tested this experimentally. This paper's just about to, we just saw proofs on this, where we took that antibody and its structure, we took the deep mutational scanning results that Sorel Fleischmann had to restrict the positions, and then we made many, many sequences, used that confidence measure to decide which designs to make. And here are the results. This is the binding affinity, and this is melting temperature, onset of melting, and aggregation. And these are the blue is the top-ranked mutants. This is the score coming out of DeepAb. And these are the top-ranked ones and the bottom-ranked ones. The affinity, there is a difference, but it doesn't look very statistically significant, right? There's a lot of noise. But the aggregation and the melting temperature, which seems to make a little more sense. So maybe when the model's more confident that we get a particular structure out, maybe that's something that's going to fold and be a little more stable into that structure. Yeah, I'll come back to that with some other models a little bit later. That was our first folding method. For our second folding method, we brought in these language models. So we made a model called Antibody that is trained on data from this community. We took antibody sequences, and we use masked language modeling. So what you do is you take a position and you blank it out, and then you ask which of the 20 amino acids should that position be, and you have the model predict those probabilities, and you reward it in training for predicting the correct wild-type sequences. So we've done that, and then interestingly, it comes up with long-range, even though there's no structure in it, it pops out long-range interactions like disulfide bonds. You see a tension between those residues, and you see secondary structure signatures in the attention matrices. So how do we do this? We used a pile of data, again, from this community and from Charlotte Dean's lab. The observed antibody space at that time had 1 billion unique sequences. We took about half of those based on some redundancy criteria, and we had an 8-headed BERT model, screening out, masking out about 15% of the sequence and asking it to reconstruct those sequences. So the way this works is you put it through layers and layers of the neural network, and then at the end you predict the sequences. If you take the final layer where it's represented, each residue in the sequence is a big vector of numbers, say 512 numbers, you kind of have a sense of the meaning of that amino acid in its context. And so you can pull that vector of 500 numbers for each residue, you can pull that out and you call that a final hidden representation or an embedding of the model, and you have some kind of representation of that sequence. So for our next generation model, we took the sequence, made antibody embeddings, took the sequence embeddings and the attention matrices, which is saying what residues are talking to what other residues, and we put that into a flow that uses a lot of components of AlphaFold. This is a 1D and a 2D track here, and then here we put a 3-dimensional track in with a template structure. We use AlphaFold's invariant point attention, which is a multi-track transformer-based network with cross-attention between the 1D and the 2D and the 3D. I like the 2D because that's like in DeepAb where we have physical residue-residue interaction terms. And then the 3D, of course, is reasoning over coordinates. We predict a structure. We take the sharp edges off with Rosetta. And then you can also take this and predict the error, how much error is in the structure. Okay. To train this, you need good training data. There's about 4,000 unique good quality structures that we took out of SabDab. That didn't seem like enough, so we augmented this data set. We did this by taking both paired sequences and unpaired sequences and making synthetic structures, and we did that shamelessly with AlphaFold. And then we plugged those structures into the model, and that makes things a little bit better. Not a lot better, but a little bit better to have more structures. When we give it a template structure, we corrupt those structures, and we only give it a template structure half the time, so it learns from the templates, and then also is able to make things without templates, and we get an error prediction out of this. The loss is where the coordinates are the bonds right, and are the atoms in the right places. This is a comparison with a bunch of other deep learning methods. The canonical framework and canonical loops encoded by the V genes come out nice and beautifully predicted, as they have in a lot of homology-based methods for a long time, because there's canonical structures. And then the H3, of course, is the most difficult residue to predict. And so you have errors in the range of 2 to 3 angstroms and longer, especially for longer CDR regions. My student didn't want to put any of the classical methods on here, but in an older paper, we had the classical methods here. This is Rosetta and A-Body Builder, and you can see the deep learning methods considerably bring this down from 3.5 to 2.5 angstroms. So we get more accurate structures out of these deep learning methods. The deep learning methods, one of these is AlphaFold, and some of the other recent deep learning methods are up here, are all kind of in the same range. So it seems like data has brought us closer to being able to predict closer to the data. But still not perfect. It seems like there's a little ways to go, and a little bit of a question of what accuracy should you expect if we had the perfect program, what's reasonable to know. So here's some examples. We get, with IG-fold is what we're calling it, we get different structures than AlphaFold. So you can see here's a case where the two structures are a little different and AlphaFold is getting it right, and here's a structure where they're different and IG-fold is getting it right. Here's the the distance to correct by the two methods, and you can see we kind of split the difference. So if we really want to know the answer, we run both, and we look at the confidence for both, and maybe use both of them in downstream docking tasks. Our method's very fast. We don't have the multiple sequence alignment, and it's a smaller network, and so we can crank things through really fast compared to other methods. Because we can do that, for kicks, we took 1.5 million sequences of paired antibody sequences, and we cranked them all through, turned the crank, and we made a resource of 1.5 million predicted structures of antibodies, for what that's worth. They're just predicted. They may not be right, but they're annotated with their confidences, and we could use this for further machine learning tools. Okay, so I told you earlier about using Sorel-Fleischmann's data and trying to predict, can it bind or not? So so this is the earlier flow I showed you. We did this with IG-fold, thinking it would be better, and it's worse. This is correlations. This is the area under the curve. And then we ran everybody else's tools also, protein MPNN and antibody MPNN and immune builder, and they're all about the same. It does seem there is some general piece that these machine learning models on antibody sequences and structures learn toward predicting affinity, even though the antigen is not in the prediction at all. But for whatever reason, IG-fold is missing that. It could be because we're predicting RMSDs. It could be there's something else weird in that, and we're trying to chase this down. Okay, could we produce new antibodies? How about generative language models for producing useful antibody sequences? Okay, so in Antibody, we took the full sequence and we did masked language modeling, meaning you take certain positions and mask them. To generate sequences, you do something different. You've seen this paradigm with ChatGPT, where you give it a question and it spits out the sentence that follows that question, or you give it a few words and it completes the sentence. So this is that GPT type model. We're going to give it antibody residues and then ask it to predict the next residue as a probability of the 20 naturally occurring amino acids. So that's fairly straightforward. One of the things we wanted to do was not just generate complete things, but be able to target, say, the H3. So, let me go back. So this is a generative model. So you could generate sequences prompting that question I opened with. If I gave you a sequence, is it real or not? And so recently, partly motivated to speak to your community, I found this paper from a few years ago where Marcoux and Mora and Walczak for Igor took an actual kind of physical probabilistic model, the V genes, the J genes, and the D genes, and then they say you can look at the probability of a scenario being the probability of all those events happening together with insertions or deletions. If you take a repertoire and train for these probabilities, then you have a generative model because you can just roll the dice for each of these probabilities and spit out a sequence, right? So that's what we want to do, and I haven't had enough time in my lab to compare with this model, but I'm curious when this thing spits out stuff if that actually looks like a real model too or not, and how so. So this is our model. It's called IGLM. Again, we're going left to right. One of the tricks we do is we formulate it as a sequence infilling task. So here we take a sequence and we take a section in the middle of the sequence and we mask it out and then give it the rest of the sequence and then ask it at the end of the sentence to put in the missing letters. So it'd be like, say, giving ChatGPT a sentence with a blank and then asking it at the end to fill in what goes in the blank. And so this will allow us to blank out H1, H2, H3, whichever parts of the sequence. So that's what we've done, and we can spit out lots of sequences. We condition this on whether it's a heavy or light chain, so it should be able to generate something that looks like one or the other, and we also condition upon the sequence, on the species. So then some of the simple consistency tests we could do is, well, if we generate, do we get how much? Do we get just the germline sequences back, or do we actually get mutated sequences? If we do this at low temperature, meaning we always pick the most probable amino acid, then we get the V gene almost 100% of the time, but we can turn up the temperature and force it to pick, when we're doing the sampling, pick it from lower probability buckets, and that will force it to get different mutations. So we can control that mutational load. As I change that temperature, and then I go back and say, well, what would the structures be if I gave it to AlphaFold? At low temperatures, I get antibodies, and then as the temperature goes up, I can get AlphaFold to throw it apart. So clearly, if we get too many mutations, it's not probably going to fold like an antibody anymore. I can also say if I generate a human heavy chain, is it really a human heavy chain? One way we checked this was with the AIRRC and just asked AIRRC what species is this and what chain. And humans, we do really well. Here's the heavy, the heavies come out all heavy and the lights come out all light. For mouse, a lot of times it thinks that the light chain is a human light chain. I'm not sure if that's what that's about, but maybe it has to do with some of the V genes, and I'm curious of these reference repertoires of the gene segments that are used to build things up. And on other species, even on some species that have little data, sometimes we get fairly accurate predictions of the light or heavy coming back, and which species. What else did we do? We can generate things. So we took therapeutic sequences, and we took their H3 and chopped them out, and then generated new H3s just to make libraries. So for each of 49 antibodies, we made 9,000 infilled sequences with different temperatures and nucleating probabilities, and we made lots and lots of sequences, and then we can make lots and lots of structures. One thing, because we're tacking on letters at the end, we can get variable loop lengths. Whereas with the masked language modeling, you're masking things and then replacing those residues, but it can't change the length because we're tacking on the end until it says end of loop signal. We can get various lengths. So we do get various lengths, and some frameworks tend to have shorter loop lengths, and some frameworks tend to have longer loop lengths. And you get a variety of shapes and presentations for possibly recognizing antigens. What else could you do? Maybe we could use this to generate sequences. We try to ask, are these sequences any good? Will they fold? Or could you try and make more developable sequences? I'd love to do this experimentally, but that's expensive, so we're still talking to people about, but computationally you can predict, is the protein going to aggregate? Here's wild type, and these are less aggregating, and you can see there's a bias toward things that are less likely to aggregate from starting with the wild type. Here's solubility, again, predicted, computed in the computer, and you could put both of those, and then you could select, say, sequences that look like they should be better behaved versus sequences that are less well behaved. And like I said, experimental testing is needed, and we've been talking to partners about this. Speaking of developability, we've in thinking about doing predictions like that, we said, well, are there dry data that we can use to say if we made a sequence, do we know if it behaves well? Does it fold? Does it stable? Does it express? Does it, does it have high viscosity? There's tons of publications in the literature where people have engineered antibodies for these kinds of properties. We tried to pull all of these together in a database calling Fitness Landscape of Antibodies, which I thought was a terrible name because FLAB just sounds kind of weak, but also considering what we're getting out of this, I think there's a lot of work to do to build our muscles on this stuff. So there's something to do. So if you look at, there's only a few studies where there's publicly available data, and they have massively different amounts of data points in these studies. We probably haven't found them all. But they're all collected by different people at different times, sometimes with different methods. So we've kept each of the data set in different publications separate so we can see can we correlate those fitness values. There's a zillion claims in the literature that we have this language model and it should predict stability, it should predict expression. And so we wanted to test that. We took language models both for antibodies, our models, as well as language models for proteins in general, and we would plot the likelihoods from the models versus the fitness measures, stability, binding, whatever. When we started this, that's what people were doing and claiming. Now everybody says, well, of course you should fine-tune on the data. If you want to make a better trastuzumab, you should have trastuzumab data and fine-tune on it, and we know that works. There's been a few publications on that. But I'd really like a general tool, so I don't have to collect all the data on my particular antibody. I'd like a general tool that works. So these are just the propensities, and you can see this is the average of all these different models and the range, but a lot of these are just not correlated. The likelihoods under these different language models don't seem to correlate to aggregation, binding affinity, expression, immunogenicity. For physics, to compare with physics, this is Rosetta, which often also doesn't correlate, but here thermostability, Rosetta does tell you something about stability, which is encouraging since it's supposed to be calculating energies of proteins. So I think there's a bit of work to do on this. We're still curating data sets. If you have data sets, we'd love to have this contributed. We'd like to, maybe this would be a good partnership with Victor's new thing too, of a public data space of things that should be available for people to test against. Okay, to finish up, how much time do I have? I want to speak a little bit about docking. 15? 13, okay. And questions, great. So I'll just go quickly through docking. So in docking, canonically, I start with two structures and put them together. It's different than what AlphaFold does, which just takes two sequences and puts them together. But I think the docking problem is still interesting. And in an antibody, I usually know the structure of, and an antigen that somebody cares about, you know the structure of, or I can ask AlphaFold. So we're still working on this start from two structures and put it together. And it's a good problem for me to test these different machine learning methods and see what they do. I'll start just briefly with the last Rosetta method that we developed. We took so Rosetta has implicit solvent and is based on sampling lots of conformations and scoring them with some kind of energy. So the last thing we did was we added replica exchange, which lets you have a hot replica that moves things around really fast and a cold replica that sinks in. And you'll see the blue ones here, you can see the backbone moving a little bit and trying to induce a fit, a very careful fit with the binding partner. And this worked really well. We thought we had the best docking method ever before AlphaFold 2 came out. So if you can't beat them, you join them. We took AlphaFold 2 and said, how could you combine these? Because we know AlphaFold gets things wrong a lot of times. So we actually take the AlphaFold predictions. We look at the confidence values, particularly at the interface. And AlphaFold often knows if the interface is wrong. And if the interface is wrong, we go back and we do a global search with our method. If AlphaFold thinks the interface is right, we still kick in our method, but we do a refinement near the AlphaFold binding site and let it move that backbone and get a nice tight interface. So when we do that, we get nice tight funnels, and these are some examples out of CASP 15, that was 2 years ago, where AlphaFold here's a nanobody binding to an antigen receptor, and it's wrong. This is the correct position, and our method, we can get the correct position. And that's true with a whole bunch of the nanobody targets that were in CASP last year. So CASP 16 is going on now. We'll see if we can do this blindly or there's other methods that claim better docking on antibodies, including AlphaFold 3. So we'll see what happens. Okay, so if we compare head-to-head AlphaRed to AlphaFold 2, now we start with the AlphaFold 2 structures and try to improve them, but we consistently have better structures closer to targets. So I think this pipeline, it takes a bit of compute time, because unlike AlphaFold that just shoots through a pre-trained method, we're searching conformations with multiple replicas running. So it does take a little compute time, but we get better structures in general. And actually we have an automated bot in the server CASP challenge that's running now for CASP, and we'll see if that works okay. My batteries might not be great. Okay, so what else? To go to straight deep learning methods for docking, we've been playing with these diffusion-based methods, like the method that made the image at the beginning of the talk. So you take an image and you add noise, and at each stage of noise, you learn to come back one tiny step to less noise, so that at the end of the day, you have a network that can take total noise and go step by step and make you a beautiful image, or in our case, make you a docked protein-protein complex, and you add noise to it, and you learn to come back and hopefully find the right docked configuration. You do this with a stochastic differential equation. You change the coordinates x with some kind of noise term, and then when you come backwards, you learn the gradient of the probability at that time step to go toward a more likely conformation of the proteins bound together. And what I like about this model is you can play with what noise you put in. So you can choose what kind of noise, how do you mess it up, and to bring it together. So people have been doing rigid body docking for a long time. I would say it was kind of, people figured rigid body was kind of solved 20 years ago when I started my professor career, and the real money was in moving the backbone. But it turns out these AI methods are not even able to do rigid body docking, so this is a nice little playground, because there's 3 degrees of freedom in translation, there's 3 more in rotation, or I do all 6 together. And yeah, these are actually reverse forward diffusion, taking the structure and noising it, so that's not our prediction. They're not quite that good yet. But that's what we can do. We train it, and then we make 400 samples, either with just translating or just rotating or both. And then this is showing the success rate, how many of our models, if we had a good scoring system, we took the best model out of 400, how many of them are close? These are all, these are classic docking methods, and then these are machine learning docking methods, including GeoDock is ours. And we beat the non-diffusion-based models. We don't beat AlphaFold 2, which has a lot more training in it. But this showed some promise to get some structures. And then inference is very, very fast. So with the physics-based docking method, I'm spending 300 CPU hours, and now we're doing this in less than a second. So maybe we can do something with sampling a lot. So how do these methods work? When you're learning grad log P, it's like you have some probability likelihood surface, and you're learning in all these random walkers as you noise things to go back toward your data into these local energy minima. And if you remember your statistical thermodynamics, where probability is related to your partition function, your gradient of the log of the probability is really the gradient of the energy of the system, or the free energy of the system. And I found this really intriguing coming from the physics side of things, because what it should be learning is these forces. The forces, if I'm docking and I'm just moving, if I'm translating, I should be able to calculate the force that's pulling it back to the correct docked position. So I found that really intriguing, but I also wasn't sure if that's really what the machine was learning. So we've been trying to take the diffusion models and look at it as an energy problem. So this undergrad in the lab looked at the math, and if you start here and you diffuse in, you should be able to integrate the forces along the way. An integral of the force should give you the change in energy. So maybe I could get relative energies of the different structures that I generate. So the change in the log likelihood is it's actually the divergence of this f function that the machine learns as you go over this path. And so we did that with a couple different docking cases. Here's the change in log likelihood versus the interface RMSD of the final structure. So these are structures that turn out to be well predicted. These are structures that are not well predicted. And you can see the negative log likelihood gets better as you get closer to the right solution. So that's really reassuring, because we could generate many structures, and the most likely ones are the correct ones. Down here is a comparison with a Rosetta based energy score, and you can see here I've got a narrow funnel. It's very deep, so I've got a broad funnel here where I'll bring things in from far away. This one, it doesn't feel anything until it gets close, and then it drops down, but the drop is much steeper. You have a very deep energy well. This is a different case that didn't quite work. We generated structures that were close, but the most likely ones were 8, 10 angstroms away. So in this case, it's definitely not learning the right energy or the right log probability for this particular docking case, even though Rosetta's, again, has a nice deep energy funnel, because especially bound docking is a solved problem, because everything's got to fit. It's like a puzzle. If you just take the shapes of a bound crystal structure and pull them apart, you can put them back together. It's when things move that it gets really hard. So this should be an easy problem, and the machine's not quite getting it. So those are some things we're working on, and there's still a lot to do. So I've shown you the machine learning paradigm is different from physics. We have lots of choices in data sets, representations, architectures, and optimization goals, tons of problems in antibody engineering to work on. We've just played around. Some models are more accurate and faster than physics, like when we're predicting antibody structures. The language models can reconstruct antibody sequences, and I'd love to talk with people more over the next day or two about how can I quality check those? How could I know? I want to find the holes in these models, and I know hopefully Charlotte will tell us the holes, because I know she's found some of them. And then we can make better models that hopefully are useful. And then more data are needed. I'm very grateful to this community. The more sequences are, the better. I would request, please get them paired as often as you can, because I really like paired sequences. I can't make a structural model unless I know the pairs. So yeah, I know it's a lot more complicated, but I love your paired data. The physics and the machine learning can synergize and work together, and then hopefully we can learn more about physics or assess the physicality of these methods. I had one plug to make. As we're designing proteins now with AI, and it's becoming easier, in some cases, a quarter of the designs just work. It's made us think, what are our ethical obligations? What should we be designing and what should we not be designing? And when it didn't really work all the time, then we didn't really worry about this too much. But there's also lots of action in the UK and internationally and in the States about what do we do with AI to keep it safe and secure? And in the biodesign space where we are making sequences, how do we make sure things are safe? And I think in the AIRR and the immune repertoire, I think that's also something we should think about is, are we putting tools out there? Are there ways we... Anyway, so we made a statement. We had a working group. We had hundreds of people sign this statement on responsible development of AI for protein design. So I invite you to visit that responsibleBiodesign.ai, and it's still open for people to sign on. And then there are some working committees that are starting to think about, if you put a model out, should it have some safety review? Or maybe should you have some safety review when you have a proposal to build a model before you build a dangerous model? Or if we do have these models, how do we make sure the access to them is, I don't know, do we need to logging or monitoring? DNA synthesis is the critical step where it goes from in silico to experimental, and fortunately there are a number of legislative advances and an executive order in the US helping put some safety at that step. So lots of thanks to my group. Jeff Ruffalo did a lot of these. Amaya Harmokar, 2 recent graduates. Michael Chung-Yoon is doing the FLAB benchmarking. Li Xin Chu on the databases, and AstraZeneca has collaborated on those experimental results. So thank you for your attention. Happy to take questions.
Yeah, thanks. Got some questions. Slido.
Try it again. It's on now.
Push the little button on it. Oh, it needs to be green.
Hello, all those people in
Grant.
Cyberland.
Grant, try that again. So taking some questions from the Slido. Could antibodies with intrinsically more rigid CDR loops explain the enrichment in binding antibodies in contrast to antibodies with a more induced fit?
Yes, I think that's what's happening. That's a rationalization. I don't know if that's actually true. It's hard to measure the rigidity. You could look at NMR S-squared correlations, but that's an expensive thing to do. Yes, I think so.
Excellent. For the use of synthetic data, is the multi-term loss weighted by if the example was a synthetic one or a real one?
Yeah, so we can make lots of synthetic structures, and there's only 4,000 experimental structures. So what we do is we pick from the synthetic structure bucket or the experimental bucket in 1/3, 1/3, 1/3 from the paired, unpaired. And so we weight them the same, but we sample them less often.
Perfect, thanks. And just one final question then. Is there a chance that AlphaFold 2 was trained on SABDAB? If I understood, your model was tested on held out SABDAB data, but what if it was already trained on?
Totally. SABDAB is all of the structures of antibodies we know, so everything before 2021 is in AlphaFold's training set. In our data, we pull out the ones that are homologous, so, but yeah, AlphaFold is contaminated. And they made a big deal about antibodies in the recent AlphaFold 3 papers. So they probably did a lot of testing on known antibody structures and antibody complexes and maybe fine-tuning on them too. So we don't totally know.
Fantastic. Once again, thank you very much, Professor Jeffrey Gray.